and today I'll be talking to you about CSRP1 knockdown and its impact on MMP expression. So lung cancer remains the leading cause of cancer deaths worldwide due to its high metastatic potential. So when I talk about metastatic, it just means that the cancer was able to travel from its primary sites such as the lungs into secondary sites such as the liver. And so this is an adhesome. An adhesome is a structure of proteins that connect to one another to allow cells to interact with other cells or the ECM or environment leading to metastasis. So specifically, we're interested in the CRP1 protein, which matches CSRP1 gene. So because the adhesome controls both adhesion and cytoskeletal dynamics, it's a key regulator of cancer cell invasion. So disruption of this adhesome can switch a cell's mode of migration. So when cancer cells migrate, they become a mesenchymal cell, and these mesenchymal cells mainly rely on proteolytic ECM degradation as the method of invasion. But when they transition to an amoeboid, these cells reduce ECM proteolase and instead rely on cytoskeletal contractility, indicating that cells can switch their invasion from protease-dependent ECM degradation to protease-independent mechanical migration. So an important protease enzyme that plays a role in degradation and remodeling of the ECM are these matrix melanoproteinases, or MMP. So within MMPs are a family called gelatinase, which specifically are known to degrade type 4 collagen within the ECM. And so these MMPs are important in tumor progression since tumor progression is driven through tumor hypoxia. And so this can promote forced creation of new leaky vasculature, which allows for cancer cells to enter the bloodstream promoting metastasis. In our previous work, we observed that manipulating CSRP1 could influence invasion of lung cancer cells. Specifically, we saw that for A549 and H1299, CSRP1 knockdown appeared to increase invasion behavior, suggesting a potential role in regulating cell migration. So we've been interested in this phenotype caused by CSRP1 knockdown to increase invasion. Considering this, I hypothesized that CSRP1 knockdown increases cancer cell invasion due to expression alteration of ECM degrading enzymes MMP2 and MMP9. So to do so, we cultured our cells, seeded them in a well plate, fixed and stained them using immunofluorescence. Then we imaged and used cell poses and image J to analyze and quantify the intensity. Overall, we saw that for A549, CSRP1 knockdown had significantly less MMP2 expression compared to the control condition. Similar results were found for MMP9, with the knockdown having less MMP9 expression. And in conclusion, these results could suggest that CSRP1 knockdown may promote invasiveness independently of ECM proteolase, potentially through cytoskeletal contractility-based mechanisms. And in the future, I'd like to test these expression levels of other adhesion proteins since they regulate cytoskeletal organization, contributing to cell migration, which could help us better understand how focal adhesion influence cancer cell invasion. These are my acknowledgements. Thank you.